Hi everyone, today we are going to talk about topic 1.3. It's an introduction to biological molecules. And sometimes you're just going to hear these called macromolecules. So bio means that they're important for life. So the macromolecules, we're going to talk about the macromolecules, how they're built. There's basically four major types of macromolecules that you need to know about for AP biology. So carbohydrates, proteins, lipids, and nucleic acids. So let's get going. Alright, so forming macromolecules. And let me make sure. Alright, so remember what it means to be dehydrated. Being dehydrated means that you don't have enough water. Synthesis means putting together. So biological molecules are formed from building blocks, monomers. Mono means one, so that's one unit, that are linked by dehydration synthesis. So if you remember in chemistry, when you had a synthesis reaction, you're making complex molecules from simpler ones. So a dehydration synthesis reaction, we're removing water, and then we're to link two monomers and form a bond, as you're going to see in the next slide. So here, this is one example of many examples of a possible dehydration synthesis reaction. Alright, so this is one example of many examples of a possible dehydration synthesis reaction. So here you have a glycerol, and you have three fatty acid chains. So the glycerol is going to give up hydrogen, and the fatty acid chains give up an OH group. So if you put two hydrogens and an oxygen together, what do you get? That's right, water. So for every bond you form, you're removing a water molecule. And this is the example where you're forming three bonds, so three water molecules are removed. All right. Building carbohydrates. So the monomers, which are joined by dehydration synthesis to form a carbohydrate, are sugars. Here you see the formation of maltose and sucrose. And let's move my picture over there. Alright, so don't worry, you're not responsible for memorizing structures of particular sugars for the AP exam. Okay, so here we have one glucose is going to give up an OH group. That's a hydroxyl group. And the other one gives up a hydrogen atom, and they form a bond or a linkage. That linkage in a carbohydrate is called a glycosidic linkage. So sugars are joined together to form carbs, and sugars are the monomers that form carbs. Okay, next we are going to build some proteins. So proteins are formed from monomers of amino acids. So here you have an example of two of the same amino acids. You don't need to know the structure of each individual amino acid, but something that gives it away is this amine group right here. So amino acid has an amine group. I always remember because it has nitrogen there. Alright, so an OH group, and one of the two hydrogens from the amine group are removed to make a water molecule, and it forms what we call a peptide bond. So linking two amino acids is called a dipeptide bond. And this is the beginning of a protein. Next, we have the nucleic acids. And nucleic acids also use dehydration synthesis. So you take a hydrogen off of one monomer and an OH group off the other one. Together they make water, and here we're showing two nucleotides joining to form the beginning of a nucleic acid or a dinucleotide. Let me move my picture there. Sorry about that. Alright, next, building lipids. Alright, so lipids are a little bit different and a little bit more complicated. There are many different ways to build lipid molecules. One of them we've already seen, which is taking a molecule of glycerol and some fatty acids to make triglycerides. So why do you think it's called triglycerides? I'll let you think about that. When we talk about cells, we're going to talk about the cell membrane and how it's made of a bunch of phospholipids. So to make a phospholipid, you do very much the same thing. You have a glycerol, and then you have two fatty acid chains. But instead of the third one, you have a phosphate group. And that's why it's called a phospholipid. Another way you can make lipids is by making modifications to cholesterol molecules, estrogen, testosterone, cortisol, the stress hormone. Any steroid hormone is basically a modification of cholesterol molecules. So lipids are a little more involved. Alright, we also need to digest these molecules when you eat your food. So the reaction that undoes dehydration is hydrolysis. If you break apart the word hydrolysis, we get hydro and lysis. Hydro means water, lysis means breaking. So hydrolysis reactions are adding back that water molecule to break the bond. So here we see our disaccharide. We have this 2-sugar molecule thingy. Our carbohydrate made of 2 sugars linked together. Alright, and here's the linkage. You're going to add back that water molecule, and it breaks that bond, and then we have 2 separate sugars. So we can do the same thing. You could break down proteins and lipids and nucleic acids the same way, all by adding water back through hydrolysis. We're using water to break that bond. Alright, let's review. Dehydration synthesis builds macromolecules. So you remove the water to join the building blocks together. Hydrolysis reactions break down macromolecules by adding that water back. Carbs are built from sugars, proteins are built from amino acids, nucleic acids are built from nucleotides, and lipids are built in many different ways using glycerol, fatty acids, and cholesterol. Alright, that's it. We'll see you guys next time. Bye.